So yes, good evening to all of you. My voice is audible. My voice is audible to all of you. Please let me know. Good evening to all of you. My voice is audible. Yeah. Okay. So good evening to all of you. And yes, today we are a little bit early because I'm not feeling well today. So I will just try to complete only a small topic today. So that is why I just came a little bit early. Just waiting for a few more students to join, then we will start today's lecture. And yes, I hope all of you have done the homework. As I am just going through your homework, your assignments. I'm just going through it. And yes, some of you have put really a good effort, and I want to thank all of you for this good efforts. Yeah. And you are even writing it on a copy with a pen. It looks very nice also. So some students are there who just send me the code PDF together. So I request you only to try to complete the assignment on the same day or maybe on the next day also. So try to keep it on the same day. Like Nancy gave me assignments 3, 4, 5 together. Kajal also 3, 4, 5 together. So it's the request only to keep it on the same day. Yes, good evening to all of you. We have Ankita, Shivam, Nilima, Yaman, Simran, Asim, Nikita, Anurima, Nivedita, Jovin. So welcome to all of you. Today I am not feeling well, so I will just take a little bit long. I'm not taking a long session today. I will keep it very simple today. And we have to discuss another technique which is there in the MBBT, which is basically SDS. So yesterday we have covered ELISA. For today's session we will keep the SDS. And I will try to complete your yeah. So I will try to complete your syllabus by this Monday or by this 15th of December. I will try to. Please, it's my request to all of you to that kindly send me the diagrams which are made by you only, not that one which you take from the net. Like some of you just take the diagrams from the net. You have to draw it by your own self. That is my request to all of you. So it is they are just taking it from the Google, I think so. So I'm not mentioning the name, but I request you to only for the diagrammatic part, only for the diagrams, that you have to draw it by your own hand. Okay, so that either it will be small or rough, any type of rough diagram you can draw, but you have to draw it by your own self. Yeah, some of you have done it very nicely. The indirect ELISA, the sandwich and the competitive ELISA. The diagrams have been drawn by many of you very nicely and very neat and clean. So yes, some students are there. We have Mridula, we have Duvin, many another also. So it looks very nice also. Then we have Harshita. Harshini, sorry, it's Harshini. So I request only to draw the diagrams by hand only. So now we will start the session of today. We have to cover FTS today. Okay. I hope many of you are joined now. So yes, Akanksha, good evening. Now I'm just going to start the STS today. Now let me show you the syllabus again. So we have covered these basic principles. Cell culture is there. In molecular biology techniques, we have done ELISA. SDS will be also there. We have done recombinant DNA technology on the weekend. Protein separation purification has been done. Electrophoresis we have done. Blotting we have done yesterday. And now the things which remain are this flow cytometry, bioinformatics, and the important part is microscopy. Applications also be done along with the recombinant part. Even I sent the PPT of these applications to all of you. Now the only thing which we have to cover is flow cytometry, microscopy and bioinformatics. But before I will take another molecular biology technique which is SDS, because SDS is also in SDS also there is separation of a protein, but some important points are there. And any one of you, have you practically done SDS? Any one of you? Please let me know if you have done it before also. Yeah, good evening Kasturi and Jaguriti. Good evening to both of you. So anyone have done SDS in their labs in colleges or institutes? Or you all are new to SDS? I think many of you must have done it also in your lab. Please let me know. Yes, a team has done it. Okay, good, good. And then the next one. Okay, Muskan, no. Dhruvi, no. SDS page has been done by John in his dissertation. Okay, very good, good, John. Some of you have done it. Okay, so I will just focus on the basics as you already know. I will let you know everything which is there in the SDS, how to operate it. But again, the main thing is that you will able to do it. You will able to understand it only then when you will do it by your own hands. Okay, so I will try to do my best to let you know about SDS. What is basically SDS page? It is similar like electrophoresis, but yes, there are lots of difference between both these two. So, okay, I will start the FPS page from the very basics. So, SDS page is, we have to discuss is SDS page polyacrylamide gel electrophoresis. Basically, we have to discuss page today. So, page, SDS is the salt. Basically we have to discuss this polyacrylamide gel electrophoresis. Like we have discussed horizontal electrophoresis, this is also the electrophoresis. So we have to discuss phase today. Now what is SDS page? So it is mostly used for analyzing protein mixture qualitatively. So when there is a mixture of proteins, at that time we need uh, the separation of proteins will be there by using this SDS page. It is useful for monitoring protein purification, as the separation of protein is based on the size of a particle. So the proteins will be separated according to their size. So can also be used to determine the relative molecular mass of a protein. So if we want to understand or we want to know the relative molecular mass of a protein, at that time also SDS page is quite useful. Now first of all we have to focus on SDS. What is SDS? It is a negatively charged detergent, sodium dodecyl sulfate. So SDS is basically a detergent, a chemical which is negatively charged, and it is used to denature and linearize the protein. So as we all know, the tertiary and the quaternary structure of the protein is highly complex. To make it linear, to make it denatured, we use the SDS. So it is used to denature and linearize the protein. So this complex protein will be changed into a linear form, coated the proteins with negatively charged, and this denatured protein will now carry the negative charge. So this denatured protein carry the negative charge all along the sequence. So this is the functioning of this SDS. Number one, what is the functioning of this SDS? SDS is a detergent which is negatively charged. It is known as sodium dudecyl sulfate. Number one, it denatures the protein, it linearizes the protein, and it also gives negative charge to the protein chain. So now, what is the main objective of SDS phase? It is basically for the separation of the proteins. So sodium dudecyl sulfate polyacrylamide gel electrophoresis is a technique which is widely used in biochemistry, forensic, genetics and more bio to separate and identify the proteins according to their molecular weight, according to their size. So according to their molecular weight or according to their size, we will able to identify, we will able to purify the proteins. So this method separates proteins based primarily on their molecular weights. So we will able to separate the protein part. So let us go with the principle now. Sodium dudecyl sulfate is a detergent which denatures the proteins by binding to the hydrophobic regions. All non-covalent bonds will disrupt and the protein require a negative charge. So the protein become linearized and it will carry the negative charge. Number one. Number two, a concurrent treatment with disulfide reducing agents such as beta mercaptoethanol or DT, which is dithiophenol, which further denatures the protein by reducing disulfide linkages, thus overcoming some forms of tertiary protein folding and breaking up the quaternary protein structure, because the very first step is that the complex structure, which is the tertiary and the quaternary structure, we have to make this complex structure the a simple one. We have to denature the protein first. For that purpose we need SDS, and along with SDS we have beta mercaptoethanol or BTT. Both these reducing agents also helps in denaturing the protein, because the tertiary and the quaternary structure is highly complex. That is why SDS plus beta mercaptoethanol is needed for denaturing the proteins properly. This is the very first important step in case of SDS page, because we are working with the tertiary and the quaternary structures. To make it denature, we need SDS plus beta mercaptoethanol or DTP. So the protein samples having uniform structure and charge, the separation will depend upon their molecular weight only. So smaller proteins migrate through the gel. We already know it. The smaller one will move fast and the larger one will take the time, as we have done in electrophoresis also. So the number of SDS molecules that bind is proportional to the size of protein. That is also the important thing, that how many SDS molecules will bind. It depends on the size of the protein. Thereby in the electric field, protein molecules move towards the anode and separated according to their molecular weight. So this is the thing. This is the complex protein structure. With the use of mercaptoethanol, with the use of SDS, we will get the protein which is denatured and which is negatively charged, as well as the number of SDS molecules depend upon the size of the protein chain, depends upon the polypeptide which is there. Now this is the one part which is SDS. Now the second thing is page. Now we have to focus on the page also. So what is polyacrylamide gel? Like we have agro gel. This is the page, which is polyacrylamide. I think this is the first homework I gave you. So this is polyacrylamide gel. The size of pores is determined by the concentration of acrylamide. Like in case of horizontal electrophoresis, we are using the agarose. Here we are using the acrylamide. So higher the concentration, the size of the pores will be small. Higher the concentration of the acrylamide, the size of the pores will be small. Now, that another thing which is there in case of FDS page is that in it we have two types of gel. We have stacking gel and we have the separating gel. So we are using two types of gel. Please keep this thing in mind. In case of stacking gel, we are using 4% of acrylamide. And in case of separating gel, we are using 15% of acrylamide. Now let me know in which gel the size of the pores will be small, either in the stacking gel or in the separating gel. The stacking is made from 4% of acrylamide and the separating is made up of 15% of acrylamide. So in both of these gels, in which the size of the pores will be small, in stacking or in separating, please let me know. Yes, please. We have two gels. We have stacking and separating. In which gel the size of the pores will be small? Yes, all of you please answer. We have stacking gel. We have separating gel. How the stacking gel have the small pore size? You have to read this line also. Higher the concentration, smaller the size of pores. Either in stacking gel or in separating gel. In which gel the size of the pores will be small? All of you please reply. Yeah, in which the size of the pores will be small according to the concentration. Now in which gel the concentration is more? In which gel the concentration is more? In the separating gel. Separating is made by 15% of the gel, sorry, acrylamide, and the before one is made up from the second one is 4% of the gel. Yes, I want a reply from all of you. Please reply. Yes. So yeah. Yes, yes. Uh, Parth, can you try to understand in which gel the concentration of gel is more? Yeah. So I think all of you get it. So yes, we have two types of gels. We have separating gel as well as we have the stacking gel. So in the stacking gel, the concentration is only 4%, but in case of separating, we have the concentration up to 15%. That means the pore size will be smaller here in case of separating gel. Then after it, why we using a polyacrylamide gel? The very first question which I asked you before also, because the polyacrylamide is chemically inert, it is electrically neutral, it is hydrophilic in nature, and it is transparent for optical detection also. Now this is the way how we have to prepare the gel. This is very important when we have to do it practically. Again, as I told you yesterday also, that these techniques take a little bit patience. These techniques take little bit patience for your handling only. When you will be able to handle it properly, then you will become the master of that particular technique. So when we are working on this gel, we have to clean the plates and combs first. Definitely we will always do. We have to clean the equipment first. Set up the plates on the rack. Pour the separating gel. Yes, one thing you have to keep in mind, that the separating gel will be the lower gel. So First we have to pour the separating gel. After it we have to put the stacking gel. So in the lower side, so this is a gel. Up to it we have to put the sorry up to it we have to put the separating gel, and on the upper one, just in this portion, we have to put the stacking gel. So separating gel is also known as resolving gel. So this stacking gel is on the upper side and separating gel is on the lower side. So this is the process of SDS paste. Boil the samples for 10 minutes to completely denature the protein. The very first step we have to we have to denature the protein. Assemble the gel in the apparatus. Pour the buffer solution into the chamber. Load 20 microliter of samples. After that you have to run the electrophoresis. Now this is the general. This is only the diagrammatic representation. This is a protein which is SDS coated. That means this protein is negatively charged. This protein is coated with SDS. Now we have to run this protein on the gel. So they are negatively charged. Na? The charge is same here. The proteins are now, they are negatively charged only. So these negatively charged protein, they are negative charge, they will move towards the positive charge. Now how the separation will occur here? The separation will occur only on the basis of their size. So if you are able to see the small proteins, they will move fast. The little bit large protein, they will there. After it, the larger proteins will be on the upper side. So the protein samples are having uniform structure and charge. The separation will depend upon their molecular weight. SDS treated proteins have very similar charge to mass ratio and similar shapes. So during the page, the rate of migration is determined by their molecular weight. Please try to understand that we already provide the negative charge to the protein. So they will migrate toward the positive end, but only according to the size. So small proteins, they will migrate fast through the gel under the influence of the applied electric field, whereas large proteins are successively retarded due to the sieving effects of the gel. So because of the pore size of the page, the small one will allow to move fast and the larger one will take little bit more time. This is again, SDS is an anionic detergent. We have to boil the sample along with beta-mercaptoethanol and SDS. It will denature the protein and it will provide a negative charge also. So on an average, one SDS molecule binds for every 2 amino acid residue. Hence, original negative charge is completely swamped by the negative charge. So we just provide the negative charge to the polypeptide chain only. Now these are the components of this gel apparatus. As I told you, we have two types of gel. We have separating gel, also known as running gel, and we have a stacking gel. The concentration of page is more here, and in this the concentration is little bit less. So first we have to upload this separating gel, and then we will upload the stacking gel. So there are two gels only, and then we have the buffer also. We have to load the samples in these gels. Like in case of simple electrophoresis, similarly here we have to load the samples on these gels, and they will move. Small proteins will move faster, big proteins will take their own time. So stacking gel, ordering, arranging and concentration of macromolecules before entering the field of separation. We are using 4% of acrylamide. The purpose is to concentrate the protein sample in sharp band before entering the main separating gel. So we have stacking gel, which is just the preparatory phase, and we have the running or separating gel. So running gel is the actual zone of separation of the particles based on their mobility. Because it consists of 15% of acrylamide, this gel has the main zone of separation of the protein. So pore size routinely used as 3% to 30%, which is the pore size of 0.2 to 0.5 nanometer respectively. So if you are able to see this figure, let me show you. This is again up to it we have the separating gel, or you can say running gel. On the upper we have the stacking gel. On the up of it we have to add the sample. Getting my point? We have to add the sample. And now upper, this is upper buffer reservoir. Now the samples will move from this stacking gel to the separating gel, and this one is the low buffer reservoir, the reservoir which is there. Let me show you one more diagram. So this is the movement of particles. If you are able to see how the separation of proteins, how the separation of molecules is there. We have three terms here. Let me show you. So this red one are the small particles. They will move fast as compared to blue one, as compared to yellow one. We have one more term which is here. We have chlorine ions which are present all over. Chlorine ions. Then we have SDS gel. Then we have glycinate. Let me show you in the. Let me show you 1 minute. Yes. So in the stacking gel, the pH concentration decreases. In the separation gel, the pH concentration is more, or the pH is more. 1 minute. So in case of this running gel or separating gel, the pH is 8.9. In case of upper gel, in the stacking gel, the pH is 6.7. You have to keep this thing in mind, that in this separating gel the pH is high. The pH is 8.9. In case of this stacking gel, the pH is low, it is 6.7 only. This is again the difference in these two gels. We have stacking gel, we have separating gel. The pH is low here in the upper gel, the pH is high in the lower gel. Again, this is how we just pour the samples, and then we have to run it. Again the same thing, that proteins will be denatured, negative charge is there, and there will be separation. We already done it. So in separating gel, the proteins separate owing to the molecular sieving properties. Smaller particles move fast. We already discussed it. So what is basically polyacrylamide gel? So polyacrylamide gel is formed by copolymerization of acrylamide and by cross-linking by N-methylene bisacrylamide. To polymerize the gel, a system consisting of ammonium persulfate and tetramethylene ethylene diamine as a catalyst. So we are polymerizing a gel using ammonium persulfate and TEMED. So this is how acrylamide along with this methylene bisacrylamide, we are just making this polyacrylamide gel. This is only like agarose. This is only similarly like agarose. This is the acrylamide gel only. But the thing which you have to keep in mind, we have first gel which is our stacking gel, in which the pH is around 6. In case of separating gel, the pH is around 8. Now why I told you this pH, I will tell you. So just keep it in mind. Just keep this thing in mind. So this is the sample preparation during this gel electrophoresis. That in the sample, like suppose I just take 40 microliter of protein. Okay, the sample like DNA isolation. I just take the 40 microliter protein sample. In this protein sample, I add the buffer, then boil the mixture for 3 minutes at 90 degrees Celsius. Now what is this disruption buffer? Basically in the protein, I will add SDS, I will add HCl, glycerol, beta-mercaptoethanol and bromophenol blue. So this is the disruption buffer which we have to add in the protein sample. So the protein sample along with the protein contains SDS, contains chlorine, that means HCl. So chlorine which is there in the diagram, it came from here. If you are able to see, here there is chlorine, there is chloride ion, so it came from this buffer. So this buffer contains tris HCl. We have glycerol, we have beta-mercaptoethanol, we have bromophenol. So when you are using the sample, you have to add, along with 40 microliter of protein sample, you have to add 10 microliter of this buffer. So Now we have to prepare two gels. First we have prepared the sample which we have to load. So this is happened of two. We have just load the sample. We have to take the sample from it, which consists 40 microliter protein and 10 microliter of the buffer. Now we have to prepare two gels. We have to prepare separation gel, as well as we have to prepare the stacking gel. The stacking gel is the very first gel in which the pH is around 6. Getting my point? So this is the stacking gel. The stacking gel again consists of chlorine. The pH is 6.8. You have to control the pH. This is the very first important step in case of stacking gel. You have to keep the pH around 6.8. So this gel contains pH trisacil with pH 6.8, acrylamide solution, water is there, SBS is there, ammonium persulfate is there, and TEME is there. So this is the stacking gel. And what about the separating gel? The separating gel will also contain trisacil but with a pH of 8.8. So again this is again trisacil, again this is also, but the pH is 8.8. Acrylamide, definitely we have to add the acrylamide, like agarose we have to add acrylamide. Water is there, SDS is there, ammonium persulfate and TEMED. So the only difference between stacking and the separating is the pH of the trisacil. Okay, so we have prepared the gel also. After it, for running the gel using tris glycine buffer. So this is the buffer which we have to use during the, when we have to run the gel, we have to use this buffer. Like in case of electrophoresis, we are using TAE, like tris acetic acid. We are using TBE. Similarly, here we will use tris glycine buffer. It also contains HCl, glycine, and SDS. So this buffer is also needed for running a gel. And then again we have to stain the gel. For that we need chemistry blue dye, and for de-staining also we need some reagents like glacial acetic acid. So this is the chemistry blue dye gel. After washing you will get these results. So these are the preparatory phase. Basically we have to prepare the sample. Number one, we have to ready two gels, stacking gel as well as separating gel. We have to prepare the buffer also. Then we have to prepare the stain also, and after it we have to de-stain it also. So these are the 5 steps which are there in case of SDS. These are the 5 steps. Now the same thing is there. We have to use SDS phase. We have to just take the proteins which are highly complex. By using SDS we are making it denatured. They become from the complex they become simple one, and then we have to pour the sample. Now this is 40 microliter of protein plus 10 microliter of disruption buffer. So we are adding the sample in the wells, and now then we have to load, and then we have to run it. So this is only simple diagrammatic representation. After it we have to visualize it under UV light using chemistry blue and for the dye purposes, and silver stain is for basically de-staining. Visualization is easy. So this is the type of uh, band which we will get. This is the type. So this is 20 kilodalton protein. This is smaller in size, so that is why it came early. And this is 150 kilodalton in size. It is heavy in size, so it takes its time and it is not moving that much faster. So now again the applications of this SDS phase we all know. I just let you know about the things which are there in SDS, the steps which are there in SDS, the things, the reagents which we need. So after it we have why we are using this SDS, because to determine the purity, to determine the molecular weight, like we will run these samples along with the molecular marker, so we will able to know the weight of a particular protein segment. Like this is of 15 kilodalton, so we will able to determine the molecular weight of a particular protein segment. Identifying disulfide bonds between the protein, quantifying proteins, blotting applications, like in case of Western blotting, SDS is must. There are some advantage and disadvantage. You have to read it by your own self. These are the applications, that is, it is for measuring molecular weight, peptide mapping, protein identification, everything which is related with protein. We have to use this SDS only. Now these are the types of gel, these are the types of results which we will get. But again, I told you that this technique is very much time consuming. When we are using fluorescent stains, then we will get the shining band. Under the influence of UV light, we will get the shining band. Otherwise, when we are using silver stain, we will get these types of results. With the help of chemistry blue, we will get blue color band. Otherwise, with the help of silver stain, we will get these types of band. These are the applications. This is a conclusion. But one thing is left here. This is not a complete thing. Um, yeah, this is not a complete thing. There is something which are left, like how, why, what is the why there is there are two pHs here. The pH one is the 6 approximately in case of stacking gel, and in the another the pH is around 8 in case of separating gel. So I am not finishing it right here. I will let you know tomorrow also regarding this SDS. But yes, you have to know the 5 steps which are there in this SDS, because the PPT is just finished. Otherwise, few points are there which has to be discussed. So I will discuss those points tomorrow. I want to discuss this stacking and separating gel more. My main focus is to discuss this stacking and the running gel more. That is why there is a pH difference in between these two gels. And what is this? What is the functioning of this change in the pH? And that will come under the another topic which is known as isoelectric focusing. I hope many of you will know it, which is also related to this SPS. So I just want to focus right now on this thing, stacking gel and separating gel, why there is a change in the pH. Okay. Otherwise, rest everything is completed. You can go and you will read it by yourself. These are the reagents which we are using. But yes, one part is left which I will let you know tomorrow only, not today. So is it okay for today? Are you comfortable? Are you getting something? It is very basic. It is very basic. So yes, I know all of you will be curious regarding this SBS, and I will suggest you one more thing to go by your own self. Even you have to watch the videos which are there in the YouTube related to SBS. I have those videos also, but you can, I think you can watch it by your own self, because these are on the YouTube only. Now the homework which is there, the again the same thing. You have to draw you have to draw this SBS page. You have to draw only. Yeah. You have to draw only the this SDS page. Otherwise, I will let you know about the difference between stacking and separating gel tomorrow. Not today. I am not feeling well today. Actually, I just don't want to take the class. Then I think let's take on a small topic. So I will let you know the one thing which is left in this SDS page, which is isoelectric focusing, which is why there is a need of two gels basically. If I completed it today, then definitely I will give you this assignment, but now the assignment is only to draw the structure of SPS only. I will let you know tomorrow why there is a need of two gels. That means why stacking gel and separating gel is needed in case of SPS. So is it clear? Are you getting my point or not? So you have to draw only. That is the thing. You have to draw SDS, not SDS. SDS is a chemical only. You have to draw SDS phase. Basically, you have to draw the structure of lactoborophase. So you have to draw SDS phase. So that's, I think that's all for today. Sorry for the small session today. So we have covered another technique which is there in molecular biology. Also, isoelectric focusing is not there in your syllabus, but it is related with the SDS space, so I will let you know. Tomorrow we will also start with another technique, which is flow cytometry. So tomorrow be ready with flow cytometry. I will try to complete the flow cytometry tomorrow. Okay. So in the meantime, in yes, you have to draw only the gel which is there, the the comb, the wells. Yeah, you have to physical figure of the places. Yes. Yeah. I will check your abstracts by tomorrow, because tomorrow I am free. Tomorrow is Saturday and Sunday. I have a holiday from my university, so I will check it tomorrow. So then I will reply you all on your mail. Yes, thank you, Shafaq. Uh, yes, tomorrow is Saturday, so I will try to take a little bit more time of all of you if you allow. Maybe we will take the class in this night session, but it will be more than 1 hour. It will be more than, it may be 1 to 20 minutes, 20 minutes more or 30 minutes more. The timing will remain the same, but maybe instead of 9, we will start by 8:30, or maybe we will start by 9 and we will keep up to 10:30. So that is the only thing. The timing will remain same. Maybe the changes will be only half an hour before or half an hour after, maybe. So this is my email ID. Maybe we will try to take it by 8:30, or maybe we will, if we will take it on 9, then we will do it for 10:30. Okay. So if we will take it by 8:30, is it okay or not? Yeah, the timing will remain the same, but it will be 20 to 25 minutes more, because I will try to complete 2 topics tomorrow. Okay. So I will share the timing, because I don't know when we will start, but I will try to keep it by 8:30, or by 8:30 or 8:40, so that you all will be free by 10 or by 10:30. But the timing will remain same on this particular evening. So I hope we will compile it on this weekend. I hope, but I'm not sure. So till then, take care of yourself. And also, I have to check your assignments which you send me today. So I have lots of work to do. So pardon me for today. We will meet tomorrow. Till then, take care of yourself. And good night from my side also. We will take by there's so much answers from for 8:30 or 9. Maybe I will take just in between 8:30 or 9. Maybe, I don't know. We will try to keep it simple. Don't worry. So that's all for today, and take care of yourself, and please try to complete your assignments on time. Then we have to focus on the review paper also. Okay. So I will reply you by this Saturday or Sunday. After it, you have to continue. You have to start your review paper also. And one more thing which I want to tell all of you, that kindly search the journals, only 4 to 5 journals which should be there in the list of SCI or UGC or Scoopus list. The journal should be free and it should be related to your topics. I hope you get it. I don't know. So you have to search the journals related to your topic. In the journal should be in Scoopers or in UGC or in SCI list. Getting my point? Let me write it. The journal should be in Scoopers or in SCI list or in UGC care list. So anyone, the scholar which is here, kindly help the another students if they will ask any question in the group. So you have to help them. What are basically these SCA scoopers or UGC care list? So they should be the journal should be in these in any of these. Either your journal, your selected journal should be in SCA list or in the UGC list, and it should be free of charge also. Okay, so we have not to pay any money for publishing our paper. So journal should be free of charge also. So anyone, if he or she has some knowledge regarding these things, kindly share your views when the students will ask any question in the group. So you have to reply it, because sometimes I am not able to reply. So I just request you to help the students, because they all are new to this field. Also, I will definitely help, but till they must have some questions regarding all these things. So anyone who has submitted any review paper or research paper before, please reply me. Anyone from you whose papers gets published, or who is the scholar, anyone from your which are present right now. John, are you here? You just said you have submitted your dissertation. Have you published any paper? Any one of you, have you published any paper before also? Because students will ask some questions. That is why I just want to select any one student or two students. Anyone is here? Please let me know. So you have to select the journals which should be related to your own topic. I'm just waiting the reply, but no one is replying. Anyone is here who has published his paper, or who has done dissertation or some type of, pursuing PhD or something? Anyone from you? I think no one is here. They are not replying. So no worries, I will help you all. Just keep this thing in mind. I will let you know later on. Okay, no worries, no worries. Don't worry. I will help you all. Uh, but I just want to let you know about these things after Sunday when I mail you. But now it's okay. I just share with you. I will let you know personally also. I will also when I mail you, I will let you know personally also. In the meantime, like on these weekends, on Saturday and Sunday, you have to go to the YouTube, and you have to, there are so many channels. You have to go for, like you have to type on Google or on YouTube that what is Scoopers, what is SCI list, what is UGC care list. Yeah, yes. Thank you, Shagesh. So you have to type it only in the YouTube. You will get so many answers. What is scoopers or leaf scoopers? What is UGC care list or what is SCA list? Basically, only those journals are authentic, or those journals are valuable, which will be published in scoopers, SCI, UGC list, or in Elsevier. So we have to search for those journals which are there in this list, because there are so many journals. Some journals are fake also. So we have the journal selection is very important. The journal selection is very important, and even if we have to take 5 to 10 days, it's okay for selecting a journal. The journal should be free of charge, number one. It should be in some authentic list. It should be in some authentic list. Yeah, Ria is here. So Ria will definitely help you. Ria has published her research paper in Mukt Shabd Journal. So Ria, is this journal is in SCA or UGC? It's UGC approved. Okay. So we will try to go with those journals which should be free of charge. Number one, and number two, which should be in the SCI list. Now what is basically SCI? SCI is Science Citation Index. Let me show you. So we have to go with SCI list. So this is the SCI list 2020. What it depends on the journal. It depends on your topic, on which topic you are working on. Please share, any one of you, please share your topic. So this is the SCI list journal which will be here. You have to download it. And please, any one of you, please share your topic. So this is the SCI is Science Citation Index. Any one of you, please share your topic if possible. So these are the 8,631 journals. These all journals are in the SCI list. Okay, so you have to select any journal which should be free of charges. Now it depends on your topics, on which topic you are working. Suppose if you are writing a topic related to zoology, so you can go with Acta Zoologica. If you are working in nanotechnology, you have to, like, you have, then you will go with advanced in advanced materials, advanced optical materials. So you have to search these things on which type of the journal will be selected according to your topic only. So like this acrologica is here, and then we have so many, of the list is too long. Critical care clinics, if your topic is related to medicine. Then we have European one is here, like heredity, if your topic is related to genetics, hepatology. So the list is so long. European Journal of Endocrinology. So if you have European Journal of Entomology, so you have to search some journals which should be free of charges, as well as like monoclonal antibodies. Na? So you have to write here, like you have to write monoclonal antibodies, antibodies related journal in UGC list, in UGC list. Okay, so these are the journals which are there, like this is the journal, or this is UGC approved journal. So the journal name is International Journal of Creative Research and Thoughts. The impact factor of this journal is 7, and here you can submit your paper also. So you have to search for the UGC approved journals. Getting my point? So is it clear to all of you? Like this you have to search your journals. Even you can go, you can use YouTube also for understanding more about the journals. So on this weekend, on Saturday and Sunday, try to search some good journals also. Okay, because I will have to check your review papers. In the meantime, you have to search for the journals also. Yes, so that you will able to, so that your work will be published in a good journal. But again the criteria is again the same. Your content in the paper should be good, otherwise your paper will be rejected. So your content should be good. It's my request to all of you, your content should be good. Either you take 5 or 10 days more, no worries, but your content should be good. So that's all from my side today, and please take care of yourself. Don't worry, send me your abstract later on, but try to, try to read review papers before writing the abstract. Okay, okay. So that's all for today, and good night from my side. And please be focused on this thing. Meet and हमारी ऑनलाइन लैपटॉप में लगा दी थी। That's all for today. I think all these terms are new to you. Kindly go through Google, go through YouTube also. You will get many information regarding Scoopers, UGC or SCI care list. As UGC care list, SCI list also है. So that's all for today. Take care of yourself and good night. Monodic, it would be accepted or rejected according to every journal has their own criteria. But if your content is, is not only a copy paste, the plagiarism will be checked, everything will be checked. If your content is original, if you are not just copy pasting, then your paper will be published. Many students from the previous batches, their papers are published in very good journals with very high impact factor. So no worries regarding it. Just focus on your paper that your content should be good. So that's all from my side. Good night to all. Good night, Microbial News. Good night, Shreya. Take care of yourself. Bye bye. And do the assignment also. Yes, yes. Good night to all. Take care. And any one of you, please pin the homework also in the group so that the rest students which are there, they will also able to submit the assignments on time. So please pin the homework also, any one of you. So thank you all for your patience. Thank you all. Yeah, good night, Ritu.